This talk will briefly cover the adoption of variant interpretation standards and guidelines into the CIViC data model via the CIViC assertion. A gene is associated to multiple variants. Choosing one brings up information specific to that variant, and below that, evidence items specific for the variant are listed. Selecting one of these brings up the evidence item. In this case, our evidence item supplies predictive evidence supporting sensitivity to the drug afatinib in lung cancer derived from a case study and linking back to the original PubMed evidence source. Given a collection of evidence items in CIViC representing the same variant and clinical annotation, such as, for example, EGFR L858R sensitivity to erlotinib in non-small cell lung cancer, this collection of evidence can be summarized into an assertion. The assertion is associated with its underlying evidence items in the assertion view of the user interface, and the assertion summarizes what's known about the variant and allows for a variant classification derived from systems of guidelines. Therefore, guideline-based tiering is built into the assertion, and two categories of assertion currently exist in CIViC based on two sets of guidelines that CIViC has integrated. This category of assertion integrates published guidelines set forth by the Association for Molecular Pathology, the American Society of Clinical Oncology, and the College of American Pathologists, which can be used to evaluate categorization of predictive, also called therapeutic, prognostic or diagnostic variant annotation, allowing a set of CIViC evidence items to support and underpin the AMP-ASCO-CAP variant tier and level, which are briefly summarized in this figure from the publication. Note that the system can annotate tumor variants that range from those with regulatory approval or adoption into recommended clinical practice to those associated with only preclinical laboratory findings, as well as benign and unknown clinical significance. To formalize integration of the AMP-ASCO-CAP tier and level into a standard operating procedure for variant curation in CIViC, minimal evidence item requirements needed to support an assertion of each tier have been adopted. The second category of assertion captures a system built for the interpretation of germline variants published by the American College of Medical Genetics and Genomics and the Association for Molecular Pathology. This system uses evidence codes derived from patient and population data, which can be summed together to allow for a more rigorous evaluation of variant pathogenicity. CIViC captures this system with the predisposing assertion, where supporting evidence items capture variant evidence criteria derived from literature sources describing patients, while other criteria applying broadly to the variant, such as frequency of occurrence in population databases, are also applied and summed at the assertion level, following the ACMG guidelines to generate evaluation of germline variant pathogenicity. Thank you.